And all right, so good morning, everyone. My name is Jesse, and I am with Exploring by the Seat of Your Pants. For those who don't know, we're all about bringing conservation, adventure, and science into classrooms around the world. And today, we are joined by the Schmidt Ocean Institute and the RV research vessel Falkor, who is a few hundred miles off the coast of Costa Rica in the Pacific Ocean, around the Cocos Islands, studying seamounts, some of the amazing biodiversity there, and working to try and get the data to help make a national marine protected area off the coast of the Pacific. We're joined by three groups from across North America, so I want to give them a chance to do a bit of a shout out. We've got Miss McDonald's grade 6s in Victorville, California. Hi, guys. Hey.
Hello.
Hello. We've got Miss Matson's grade 9s in Green River, Wyoming. Hi, guys.
Hi.
Hello. And we've got the homeschool group in Kitchener, Ontario, grade 9s, Van Buttes. Hi, guys. Awesome. We are joined live on the ship on the aft deck by Dr. Eric Cortez, who's going to tell us a little bit about the work that they do on the Falkor and this mission specifically. Without further ado, thanks so much for joining us, and take it away.
All right. Hi, everybody. Can you hear me all right?
Yep. Great.
All right. Good. So hi, I'm Dr. Eric Cortez. I'm the chief scientist on this expedition on the Falkor with Sebastian, which you can see behind me. We're standing on the aft deck right now, and we are transiting from one of our sites where we were diving yesterday to another site where we'll do a dive tomorrow. So, and so you're very lucky actually to see the ROV on deck. They're doing some work on it right now, but let me give them a little tour. This is actually the back of the vehicle. And these are some of the thrusters that move it around in all three dimensions, X, Y, and Z. Some of the lights on the back. This cable right here is the connection that runs into the control room, and it sends all the commands down to drive the vehicle by the pilots in real time. These big storage bays on the side that we can add all sorts of different equipment. They have two of these manipulator arms, and this is how we collect all of our samples and interact with the environment while we're down there. They have these little manipulators on the end that we can take samples with. We put those samples in a bunch of different containers. We have these little tubes that we can fill with lots of smaller samples. This thing we call a bio box in the back, which helps keep the samples nice and cold before they come up to the surface, because a lot of the animals are really sensitive to air. It's very cold down here, and especially you can see it's very hot up here in Costa Rica. And these are push core tubes, so the sub will grab one of these and we can push it down into the sediment. It kind of works like if you take a straw and put it in your drink and then stick your finger over the top and pull it up, you can pull some of the water out. Same kind of thing. So there's a little valve here. This goes up, so you push the push core in, and you get some mud in there, and then this seals, and you pull it right out, and we can collect sediment samples and all the organisms that are inside the sediments that we'll study later on. We have a variety of other little nets. This is a temperature probe if we're someplace really warm. This is a suction hose, the slurp sampler. This is a lot like a vacuum. So the sub picks this up and then sucks up any of the little critters. It's good for chasing down crabs and shrimp and a lot of the worms. And those come through this hose into a series of containers back here. And then a lot of the work that we do is really visual. And this ROV, you'll see some of the highlights.
Eric, if you can hear me, we just lost you for a second there. Whatever section you're on, it disappeared. So we'll work to get him back for a few seconds, guys. This is what happens sometimes when you're bringing hangouts from ships in the middle of the ocean, is that the connection sometimes doesn't work, which is quite all right.
While we're waiting to get Falkor back online, we could start by taking a question about Falkor, if anybody has anything.
Yeah, we certainly could. So Miss McDonald's group, if you guys have a question about the ship generally, I know we haven't dove into exactly what they're doing on this expedition too much yet, but in general, if you guys have questions about what the Falkor does, what their expedition's like, that would be awesome.
Can you? Hi, everyone. I'm Lisa Levin. I'm one of the scientists on board from Scripps Institution of Oceanography. And I guess I'm just going to fill in for Eric while we're trying to get his connection back. Eric, as Eric said, he's the lead, but there are a number of other scientists out there, of us out here. We have been studying the seafloor. It's been really exciting for us. Every single day we dive in a new place that no human eyes have ever seen before. I can't tell you what it's like to go down there and realize that you're the very first person ever to see an animal or even a whole scene down there. We've been, as Eric said, we've been studying seamounts and methane seeps, which are areas where methane comes out of the seafloor. And the life has been amazing. We've seen massive clusters of strange starfish, brittle stars on the bottom. We've seen masses of sponges and corals. What? Just a minute. Eric, tell Eric there is no connection out there. He should come back. Yeah. Okay. So Eric will be back in a minute. We're just trying to communicate with him. But we've seen massive gardens of sponges and corals. When I say garden, it means that animal life is super abundant and big. And what's amazing is some of these animals are really old, like thousands of years old down there. And to realize we're the first person to see them is pretty amazing. I'm not sure if I should keep on going.
Yeah, that's great.
Or answer a few questions from the students while we're waiting.
Well, while we're waiting, you know what? Before we get into questions, first of all, thank you so much for jumping in. One of the things that was discussed before we had classes in is that you guys found a really unusual trash pile. Could you tell us a little bit about that?
Yes, I can. It really happened only on one day. Well, maybe Eric's coming back here. But we were diving in a very, very deep spot where two continental plates come together, and it seemed like debris accumulates down there. When we made that dive, we first saw a random plastic bag or two, but then we saw a massive pile of plastic bags. So that was pretty depressing for us. Okay, I'm going to give the microphone back to Eric now. He'll pick up where he left off.
All right, thanks, Lisa. Lisa's not going anywhere, so you can ask her questions too. So I guess we got pretty far through the tour. You got to see the ROV. There was another vehicle on deck that I was talking about, I think, after you lost me, which is called the Wire Flyer, which does some oceanographic profiling, which is a really interesting piece of equipment, but you got to see the ROV at least. So I don't know how much Lisa talked about. Okay. Yeah, should we go through? Let me show you some of the map and some of the things that we're doing out here and some of the footage that we've been able to take over the last couple of weeks.
Great.
Okay, so Costa Rican deep sea connections, looking at the connections between the deep and the shallow. So this is a map looking at some of our sites offshore Costa Rica there in Central America. The sites extend from the coast of Costa Rica all the way out to Isla del Coco, which is down sort of in the southern part of this list. The sites closer to the margin are cold seep sites where there's methane coming out of the seafloor. And then the seamounts are sort of like stepping stones that go all the way out to the island at Isla del Coco, and actually continue all the way out to the Galapagos. They're all connected together. So we're looking at the seeps on the landward side of the subduction zone. So the Cocos Plate, which is further out in the ocean, is subducting or sliding underneath the Caribbean Plate, which is where Costa Rica is. And it's the seamounts in this chain that are actually moving underneath the plate and causing the seepage that we see on the continental margin. So they're pushing all the fluids up to the surface, and they're coming out, and their surface expression leads to a lot of methane and hydrogen sulfide and some really cool communities that live off of that chemical energy. And then the seamounts, we're looking at those to see how connected the shore is to Cocos Island National Park further offshore, and to support some conservation efforts that are aimed at trying to protect some of those seamounts, which we've been discovering are really unique habitats and very different from anything else that we've looked at so far. So here's the RV Falkor. You got to see us on the back deck of the ship just a minute ago. This is Sebastian in the background, which you're now familiar with, and then the Wireflyer vehicle in the foreground. And this is oceanographic profiler. So there's a big weight that you tow behind the ship, and it flies up and down the cable that's attached to that big weight, clump weight at the bottom. And it has a whole bunch of sensors on it. And as it flies up and down, it gathers a whole bunch of data in the water column, so we can get this really beautiful two-dimensional profile. And last night and early this morning, we used it with a bunch of acoustic sensors that were on it, so we could see what's called the deep scattering layer, which is a whole group of fish and plankton that spend the night up at the surface in relatively cool waters in the dark. And then as the sun rises, they start to swim deeper and deeper down hundreds, or even in some cases to 1,000 meters down, where there's no light and the waters are much cooler. So they can conserve energy and they can avoid predation. So we basically watched that happen this morning, which is really, really cool to see. So there's Sebastian being launched, which we've done, I don't know what we're up to, 18 times, I think, on this cruise so far. And that's us in the control room. That's Lisa and I and the pilots in the background there running a dive. We talk to the pilots and explain what we want to do, and then they fly the vehicle and control the manipulator arms and collect all of the samples for us. And we work together with them to figure out the best way to do that with all of the different tools that we have available to us. So let's show you some of the highlights of some of the dives that we've done so far. I don't know if you guys have checked out the website, but you may have seen a couple of these.
And we'll pass the website to all the classes at the end too, so you can see even more when we're done.
Perfect. Are you guys seeing the video now?
Yes, we are. It's awesome.
Okay. So you're seeing video from a couple different places. In some places there are the seeps like this, these tube worms, which live off of hydrogen sulfide that's present in the waters and the seeping fluids that are coming out. They're entirely reliant on entirely reliant on the chemicals that are coming out of the seafloor. For all of their energy, they basically pass those to bacteria that are living inside their tissues, and they make all the food for them. And then further out on the seamounts, these really remarkable coral and sponge communities that we've found, that some of these we found just yesterday. And we were really surprised at some of the species that were there, and we started to describe the different zones on the seamounts. Just like in the intertidal zones, you would see areas with different temperature or different oxygen levels, really changing the kinds of communities that we saw there, from corals to sponges to these just fields of little brittle stars that were in some of the areas that are really low in oxygen, and really just some of remarkable habitats. We were staring at one of these yesterday towards the end of the dive and just all talking about how it felt like we were just scuba diving and on a shallow water coral reef, except we're so far down in the ocean. It's really remarkable, the beauty and the age and the fragility of some of these habitats. And so that's why we're using a lot of this information to work with our colleagues from Costa Rica who are out with us now to try and set up some conservation areas that will protect these seamounts and expand the Cocos Island National Park to include some of these seamounts all the way back to the margin to make sure that these communities stay protected and stay in the really pristine state that we saw a lot of them. I know Lisa was talking to you about some of the trash that we saw. We've also seen a lot of evidence of fishing activities. We saw a lot of long line yesterday when we were down with the ROV. And so that can do a lot of damage to a lot of these communities. So we're really trying to relieve some of the fishing pressure, move that outside of what we hope will be a national park at some point, and to try and make sure that these communities stay as pristine as possible in the future and keep their role as a fishery nursery ground, and connect the whole deep sea ecosystem to maintain its health and all the services that we get from the deep sea that we're really just starting to understand. So I think I'm happy to take questions. I think we have plenty of time. Lisa's right over here too. She can help me answer some of these.
Come on in, Lisa. Everyone join in. Outstanding, guys. That's really, really neat. Before we dive into questions, I really want to just make sure class understands. So these seamounts, this is an underwater mountain range where it leads to islands, which are like the peaks of the biggest mountains that are under the water, and you guys are tracing the journey along this route. That's really cool. All right, yeah, let's go over to Miss McDonald's class. If you guys have a question, come on up.
How far have you made it in the boat? How far, like, around have you made it in the boat?
How far? I didn't quite hear the question. Oh, how far out were we?
How far around the world
we were.
have you made it?
Oh, the Falkor has been all around the world. Over the past 6 or 7 years, they started up in the North Sea, and then they were on the East Coast of the U.S. The first time I was on the Falkor was in the Gulf of Mexico in 2012. And then they came through the Panama Canal. They've been working on the West Coast of the U.S. They were out in Hawaii for a long time. I sailed on the Falkor a couple years ago from Hawaii all the way down through the Phoenix Islands to Samoa, and they've been working in the South Pacific. And I think this coming year they go to Australia, so they're crossing the Pacific again. So they can go all over the world. We have been on this cruise a couple 100 miles out to get to Isla del Coco in the national park. And our deepest dive on this cruise has been about 3,600 meters, which is over 2 miles down in the bottom of the ocean.
Very cool. How long has this journey been? How long have you guys been out?
We left on January 6th. So we're nearing the end of our journey, which is the total about 3 weeks long.
Okay. All right, let's head to Miss Matson's class. You guys have a question?
Guys, anyone have a question?
Yeah, I just walked.
Joe, yeah, we don't have a question since we had the transition, so our kiddos don't have any
That's
questions.
okay. Cool. We'll come back. If you guys have any questions after you hear more from the other groups, let me know. All right, let's head to Kitchener. Yeah, our student there. If you guys have a question, just come on up. You should be demuted.
What do you have to study to do this?
That's a good question. Everything. We have scientists from a variety of different backgrounds. I'm primarily an ecologist, but I studied marine environments and oceanography for most of my schooling. But that includes everything, math and chemistry and physics. and lots of biology and lots of oceanography. But we also have microbiologists out here and geochemists. We have taxonomists and vertebrate zoologists who study all of the different animals that we're finding. We have engineers. We have a number of engineers. The wire flyer vehicle that you saw before was built by an engineer who's on our project team, and one of his technicians and a student are out here running it now, and they're engineers. Of course, all the engineers that work on the ROV, all of the other technicians that help us out here with the mapping and the other oceanography that are part of the team that works on the Falkor. And then we have the chefs and the captain and the deckhands and the crew. And I don't know, what was your background, Lisa?
Well, like Eric, I'm an ecologist, so I studied biology and and oceanography for my PhD. But really, if you guys want to make a career out of this, get a fundamental science background. That's how you start.
Very cool. Always like when that question gets asked. All right, let's head back to Miss McDonald's class. If you guys have a question, come on up.
What's the most unusual thing you discovered?
Ooh, wow, there's a long list. The most unusual thing. Well, something I had never seen before. You saw just a little clip of it. It looked like a giant tongue moving over the seafloor. That's from the Xenoturbellida. It's a very strange group that has only been seen a couple times in the deep sea and is very rarely collected. That was very weird. So that was really cool to see. I don't know, there's so many. We found some species of corals that I was not sure we were going to see, but we did. These are the scleractinian, the reef-forming stony corals that you normally think of as shallow water coral reefs. We found one hundreds of meters down in an area that's very difficult for corals to survive just because of the really low pH naturally, but also from a result of ocean acidification. So we were really surprised to see that. I don't know, what was your favorite?
Well, I think one of the weirdest things that we saw was a seafloor that was entirely pink because it was covered by dense brittle stars, which are little like starfish, but there were literally millions, if not billions of them, so that every single rock and every bit of sediment and everything was covered with them. And they were in a pretty narrow band, about 100 meters deep, but they were all over the place on several of the different seamounts. And they are one of the animals that can live at really low oxygen. So we think that oxygen has something to do with it. But that was the weirdest sight. The most exciting thing for me, I found, we didn't talk about our single-celled animals called xenophyophores that live on the bottom that are the size of your fist and agglutinate sediments. But one night we were dissecting them and I found a cluster of fish eggs inside. And the idea that a vertebrate would use a single-celled animal as a nursery habitat was kind of exciting.
Very cool, guys. We would also love to have that picture. If you guys have pictures of those brittle stars, that's amazing. Like, that is super cool. I wanted to ask too, so you guys, because you're studying seamounts, it's really worth noting. So on land, of course, as you go up mountains, you get different habitats, different creatures. Do you find these completely different areas when you're looking underwater?
Yeah, we do. What Lisa was just describing, that band of brittle stars, which we decided a group of stars is called a galaxy. So there's a galaxy of brittle stars. That was on a few different seamounts, and they were 100 miles apart. So it wasn't just unique to that one seamount. It was this band at the same depth in two different places. So we think that may be a sort of persistent kind of type ecosystem in the oxygen minimum zone at a whole bunch of these seamounts. And there were, when we started deep, there was a big diversity of corals. Some of them you saw in some of those video clips. And then the corals almost disappeared, and it was a band of sponges. And then the sponges were still there, but disappeared a little bit. And then the brittle stars showed up. And then there was another band above that where there were these little tiny scallops with shells that were paper thin. And then as we got even shallower than that, the corals started to come back. So it was really these different zones on the seamounts, and that was repeated on multiple seamounts. So we think that they're really persistent. Of course, every seamount is also unique, and so there were things that we saw on some seamounts that we didn't see on any other seamounts. So it's this combination. We think some of it's predictable, but also each one of those is unique and really deserving of protection on their own.
Very cool. All right, let's head back to Ms. Matson's class, see if you guys have a question now. If you do, great. And if not, that's okay.
Yeah. Okay. So what we wanted to know, or did you have a question, Malachi?
No.
Okay.
I have a question for you.
So you said that you found this, I guess, patch of garbage. What do you know why that it was collected in that specific area, or are you researching the reasons why garbage might be attracted or settle in that certain spot?
Good question.
Yeah, as I mentioned, it was very deep. It was at a place where there's probably a dip in the seafloor because one continent is moving, continental plate is moving under the continent. So we call those trenches, and we were at the very edge, the upper edge of the trench, but it looked like a place where obviously garbage could collect. We saw, as I mentioned, this came because somebody actually asked about the garbage. So, but we saw a few bags, but then a big pile of bags, and we didn't really explore extensively at that in that area. But it seems like garbage may collect in trenches all around the world.
There you go. Thank you so much. All right, let's head to Kitchener again. If you guys have another question, go right ahead.
How many people does the ship that you're on have on it when it travels?
How many people?
Yeah. So yeah, how many people on the vessel?
Okay, 43 people total. I had to get a little help with that question. So yeah, 43 total. We have about a dozen scientists, and then there's like the technicians, the engineers, the engine room, the deckhands, the officers, the ROV crew. So they're all distributed among those.
Yeah. I always like highlighting that, you know, you don't necessarily, I mean, it's great to be an established and distinguished scientist, but you can get on vessels like this and partake in exploration like this with a huge variety of skill sets. And I always like to make sure people know that in case they're not science inclined. But yeah. All right, let's head back to Miss McDonald's class. You guys have another question?
I have a question. So I just first thank you guys for doing this, because ours live in a desert and we're about 110 miles from the ocean. So this is really cool for us to see. But my question is, after this expedition is over, how long do you wait before you go on to the next expedition?
That really depends. My expedition, my next one is in April, so it's coming up pretty quick. This has been a really busy year for me. This is my third in the last 6 months, and I have another one coming up. That's, I think, abnormal. That's a lot. For the last, I don't know, 20 years, I've been going on 1 or 2 on average expeditions per year. Would you say that's about, when's your next one?
My only other coming up expeditions are student cruises at the end of the year. But over the past 35 years, I've been going one to 3 times a year, but 3 would be a lot for me. Eric goes a lot more.
Just this year. It's a busy year. It's usually 1 or 2.
Really quickly, follow-up, could you tell us a little bit about what a student cruise is and how students might get involved in something like that?
Oh, yeah. Well, my student cruises are actually field trips for my deep sea biology class. At Scripps, we are very, very fortunate to have university state support for ship time to allow us to take students on ships for classes. These are just one-day trips. Actually, I have a couple of them. We have the luxury of having the deep sea right outside of San Diego. We can steam for 2 hours and be over 1,200 meters of water depth. So I just take my classes out. That's typically 20 people. There are other opportunities for students to go, but I am less clear on how. I think there's an age limit, for example, on most ships of either 14 or 16 years old. So.
Good to know.
Yeah, but a lot of those, there are other ways to get on cruises like that. Most of them are aimed at college age students, but there are opportunities through UNOLS, which is the National Oceanographic Fleet in the U.S., and they occasionally run cruises to basically train students in how to run a cruise. So those happen. They've been happening about once a year, actually, recently. Or just, taking a course. There are a variety of courses that have labs like Lisa's describing, particularly on the West Coast where it's easy to get deep quick. But yeah, there's lots of opportunities out there.
Very neat. Thanks, guys. All right, Ms. Matson's group, if you guys have a second question.
Yeah.
What changes are you guys seeing that are accelerating?
That's a good question. Things change slowly in the deep sea. So we're really just getting enough data to be able to measure change and measure the rate of change. But even then, some things have been seen. A lot of the heat that's going into the atmosphere ends up in the ocean, and a lot of that actually ends up in the deep ocean. And the deep ocean is so huge that it can absorb a lot of that heat and only change temperature by a few tenths of a degree. But that has been measured in a few places, especially in the Mediterranean and the Gulf of Mexico and smaller bodies of water like that are connected to the oceans. And near the poles, where it's warming much faster, we've seen those changes in the ocean. We're also looking at changes in pH through ocean acidification. All the CO2 in the atmosphere makes it into the ocean. The ocean absorbs a lot of that CO2 as it comes to equilibrium. And that CO2, that gas, as you bubble it essentially through water, it turns it acidic. That's why sodas that you would drink are acidic, because of all the carbon dioxide that's in there. And that makes it harder and harder for organisms to lay down calcium carbonate shells, like all of the scallops that we were talking about before, these paper thin shells. It's keeping corals from growing in some of the areas that we've looked at. And those changes have been measured, and that is changing pretty quickly. And that's especially significant in places where the pH is already pretty low, like this area out here. There are these big oxygen minimum zones, and there's very low pH in those zones. I'm going to let Lisa talk about oxygen, but that's another one that's changing.
Yeah. It turns out that as the ocean is warming from climate change, that warmer water holds less oxygen. And so the whole ocean has been losing oxygen because of that. And also because warmer water is what we call stratified in layers, and there's less vertical mixing. And so oxygen's not getting from the air into the interior of the ocean. So we've documented, scientists have documented that the ocean has lost 2% of its oxygen overall in the last 50 years. But some places, the oxygen loss has been really extreme. And so the North Pacific is one of those. And for example, off California, where I live, Monterey has lost 40% of its oxygen at a couple hundred meters depth, and Southern California has lost about 20%. So oxygen loss is another consequence of climate change. And these areas we're studying are really interesting because there's naturally low oxygen off Costa Rica due to decay of primary productivity. And the patterns we're seeing are like a glimpse into the future of a less well-oxygenated ocean.
Right. That's fascinating, a little dire, but very, very interesting. So that's, you mentioned some of these effects are concentrated near the poles. Is there a reason why that would be?
The pH effects? Yeah. Yeah, I mean, let's think about temperature. I mean, the warming, I'm not sure about. There's something called a Ravel factor and water temperature that affects the exchange in gases and the dissolution of carbon dioxide. But do you know this better than me? Probably. So basically, in cold water, you can dissolve more gases. So I think more carbon dioxide is actually being taken up at in the polar waters, and acidification is very much more severe. And the effects of warming, of course, there are very intense and melting a lot of ice, and that's changing the ecosystems up there completely.
Right. Thank you, guys. And then we'll take one last question before we do a bunch of wrap-up elements of the talk with back to Kitchener. If you guys have one more question, yeah, take it away. If you do. If you don't, we'll go back to another class, which I think we'll do. Let's go to Miss McDonald. Oh, no more questions? Okay. Yeah, that's okay. Miss McDonald's class, do you guys have a last question?
Go ahead, go to the camera.
I heard there was like flowers under the water. Is it true? Is it true that there's actually flowers underwater?
Well, there's no photosynthetic plants in the deep sea because there's not enough light. So there aren't true flowers, but some of the sponges and some of the other animals, invertebrates down there look a lot like flowers. So when we talk about a sponge garden or a coral garden, we're not talking about real flowers and real plants. We're talking, it's a very human term. We're talking about the appearance of the invertebrates that look like a flower garden.
Cool. I urge all the classes to look up glass sponges when you're done this talk, because they're some of the most beautiful things in the entire world, and you find them in the deep sea. And if you want things that are flower-like or just incredible, that's something to look into. So guys, this has been marvelous. Is there any last messages for classes that can look into what you guys are doing in more detail? Any way you can point them to more resources, or tell us a little bit about what's going to happen once you wrap up this expedition?
Sure. There's a ton of resources on the Schmidt Ocean Institute website. There's a lot of coverage of this cruise that our communication specialist Monica has been putting up for us, and the videos look just fantastic. You can hear a lot more of the background on there. There will be I don't know, we have a lot of work to do. We collected a lot of samples. It'll take us a long time to get through them. We have a few things that we know sort of immediately, but it takes a long time to identify all the animals that we've collected through genetics and just through their morphology. A long time to count everything and come up with differences between communities in different places. We got to go back through all the video. So, you know, this one cruise will produce information and data and samples that will take us literally years to get through. So, you know, we found some things out pretty quickly, and then a lot of them just take a long time. So stay tuned. I just want to thank everybody for their great questions and their interest, and it was fun talking to you.
So fun having you guys. And for our classes, so boys and girls, I'm going to demute both your microphones. If you guys could join me in saying a huge thank you to Lisa and Eric, that would be great. So yeah, you guys are demuted. Go for it. Thank you. Awesome, guys. So yeah, throughout the year, we're going to be doing several more hangouts with the RV Falkor. So we hope you guys will join us back for those. Eric and Lisa, you guys are marvelous. Even if you guys aren't on a vessel, please do get in touch. We'd love to host you back to highlight some of the stuff you guys are doing in those several years of getting through the research. And till then, we appreciate it, and have a lovely rest of your day, guys.
Great. Thank you.
Bye.